Hi everyone, Gina Logrande here. Thank you for taking the time to view my presentation on the citric acid cycle. I hope you enjoy it. The citric acid cycle, also known as the TCA cycle or Krebs cycle, is a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl-CoA derived from carbohydrates, fats, and proteins into adenosine triphosphate, ATP, and carbon dioxide. The cycle also provides precursors of certain amino acids, as well as the reducing agent NADH that are required for many other biochemical reactions. So how does the cycle work? The cycle consumes acetate in the form of acetyl-CoA and water, reduces NAD+ to NADH, and produces carbon dioxide as a waste byproduct. The NADH generated by the citric acid cycle is fed into the electron transport pathway and is known as oxidative phosphorylation. The net result of these two closely linked pathways is the oxidation of nutrients to produce usable chemical energy in the form of ATP. In eukaryotic cells, which is the focus of this presentation, the citric acid cycle occurs in the matrix of the mitochondrion. The citric acid cycle involves 8 chemical reactions that produce carbon dioxide, ATP, NADH, and FADH2. The NADH and FADH2 are electron carriers that can be used by the electron transport chain. As we discussed in an earlier presentation, the precursor to the citric acid cycle is the conversion of pyruvate from glycolysis to acetyl-CoA, which is then oxidized to CO2. The electrons liberated by this oxidative process are passed via NADH and FADH2 through an elaborate membrane-associated electron transport pathway to oxygen, the final electron acceptor. Electron transfer is attached to a proton gradient across the membrane. This gradient represents an energized state, and the energy stored in this gradient is used to drive the synthesis of many equivalents of ATP. Oxidative phosphorylation is the term used to denote ATP synthesis as a result of electron transport. So what are the steps? On the following slides, I'll explain each one of the 8 steps in the citric acid cycle. For each step, the portion of the molecule that undergoes a change is highlighted in blue, while the enzyme that catalyzes the reaction is highlighted in yellow. In step 1, the enzyme removes a proton from the methyl group on acetyl-CoA, and the negatively charged CH2 forms a bond to a carbonyl-carbon on oxaloacetate. The subsequent loss by hydrolysis of coenzyme A drives the reaction strongly forward. The end product of this reaction is citrate. In step 2, an isomerization reaction in which a molecule of water is removed from citrate and then another is added, moves the hydroxyl group from one carbon atom to its neighbor. The enzyme aconitase catalyzes the dehydration reaction to isocitrate. In step 3, the first of 4 oxidation steps in the cycle, isocitrate loses a molecule of carbon dioxide and is oxidized, forming the 5-carbon alpha-ketoglutarate. NAD+ is reduced to NADH and H+ in the process. Isocitrate dehydrogenase catalyzes the reaction. In step 4, alpha-ketoglutarate is converted to the 4-carbon succinyl-CoA. A molecule of CO2 is removed, and NAD+ is reduced to NADH and H in the process. Alpha-ketoglutarate dehydrogenase catalyzes this reaction. In step 5, CoA is removed from the succinyl-CoA molecule and is replaced by a phosphate group. The phosphate group is then removed and attached to guanosine diphosphate, thereby forming guanosine triphosphate. Like ATP, GTP is an energy-yielding molecule and is used to generate ATP when it donates a phosphate group to ADP. The final product from the removal of CoA from succinyl-CoA is succinate. Succinyl-CoA synthetase catalyzes the reaction. In step 6, succinate is oxidized, and fumarate is formed. Flavin adenine dinucleotide, also known as FAD, is reduced and forms FADH2 in the process. Succinate dehydrogenase catalyzes the reaction. In step 7, a water molecule is added to fumarate, and the bonds between its carbons are rearranged to form malate. Fumarase catalyzes the reaction. In the last of 4 oxidation steps in the cycle, malate is oxidized, forming oxaloacetate, the beginning substrate in the cycle. NAD+ is reduced to NADH and H+ in the process. Malate dehydrogenase catalyzes this reaction. So how many ATP do we actually get? In eukaryotic cells, NADH generated in the cytoplasm during glycolysis must be transported across the mitochondrial membrane before it can transfer electrons to the electron transport chain. This process requires energy. As a result, between 1 and 2 ATP are generated from these NADH. For simplicity, however, we will look at the theoretical maximum yield of ATP per glucose molecule oxidized by aerobic respiration. In calculating our totals, we will assume that for each pair of electrons transferred to the electron transport chain by NADH, 3 ATP will be generated, and for each electron pair transferred by FADH2, 2 ATP will be generated. Keep in mind, however, that less ATP may actually be generated. Through the process of glycolysis, which we see in the image, 1 molecule of glucose oxidized by aerobic respiration in prokaryotes yields 2 net ATP by substrate level phosphorylation, 6 ATP from 2 NADH by oxidative phosphorylation, and through the transitional reaction we get 6 ATP from 2 NADH by oxidative phosphorylation. Through the citric acid cycle, we get 2 ATP by substrate level phosphorylation, 18 ATP from 6 NADH by oxidative phosphorylation, and 4 ATP from 2 FADH2 by oxidative phosphorylation. The total theoretical maximum number of ATP generated per glucose in prokaryotes is 38 ATP. 4 from substrate level phosphorylation, 34 from oxidative phosphorylation. However, in eukaryotic cells, the theoretical maximum yield of ATP generated per glucose is 36 to 38, depending on how the 2 NADH generated in the cytoplasm during glycolysis enter the mitochondria, and whether the resulting yield is 2 or 3 ATP per NADH. That wraps up my presentation on the citric acid cycle. Many thanks for taking the time to view my presentation. I hope you enjoyed it, and I look forward to viewing each of your presentations over the next few days.